Today, we're diving into what is arguably the most epic, high-stakes biological survival race in existence, the incredible mechanisms behind how we all begin. We are going to break down the entire process, everything from the microscopic, completely wild journey these cells take just to meet, all the way to the profound hormonal changes that sustain a new life. We're even going to look at how a simple little test strip detects that whole shebang. Honestly, before we're even born, a completely ridiculous amount of effort goes into just getting us to the starting line. It's this fascinating, perfectly choreographed biological dance, and we're going to make sense of every single step of it together in a really fun, intuitive way. So let's jump right in. Okay, let's just pause and think about this number for a second. 200 million. That is, roughly speaking, the entire population of Brazil all sprinting at the exact same time. How does one single cell, out of an initial starting lineup of 200 million, navigate a microscopic, invisible obstacle course to actually create new life? It is literally the most competitive race on Earth. But, before the starting gun even goes off, our two competitors, the egg and the sperm, have completely different ways of preparing for this. The starting line looks wildly different depending on which side you're looking at. For females, the preparation is incredibly selective. Actually, females begin creating their primary eggs before they're even born. The process literally hits the pause button for decades, and then, typically, just one single meticulously prepared cell is released per cycle. Males, on the other hand, it's a continuous, high-volume production line. Taking about 74 days to mature, the male body pumps out a staggering 120 million sperm every single day. So you've got one meticulously prepared egg waiting for one of millions of continuous runners. All right, let's actually run this race. Despite millions starting the journey, biological selection, sheer distance, and these immune cells called macrophages, which honestly basically acts like biological bouncers, create an extreme funnel. Out of the 200 million sperm deposited, only about 100,000 make it to the uterus. From there, a mere 200 survive the trek into the fallopian tube. And at the very end of all of that, usually only one single victor will actually fertilize the egg. It is a biological gauntlet of absolutely epic proportions. Navigating this daunting internal map takes about 5 hours, but the sperm aren't doing it totally on their own. The transport is actually a deeply cooperative process. Sure, it relies on the active swimming of the sperm itself, but it's heavily aided by passive transport. Think of it kind of like being pulled along a moving walkway at the airport. Uterine contractions, which are stimulated by specific chemicals from the semen, along with the natural flow of uterine fluid, actively help sweep those surviving sperm up toward the fallopian tubes where the egg is waiting. But just arriving? Yeah, that isn't enough. To even stand a chance of interacting with the egg, the sperm has to undergo a final, absolutely crucial functional maturation step called capacitation. Essentially, the sperm has to take off its training weights. Triggered by enzymes in the uterus, the sperm loses its outer shield, this thick cholesterol and glycoprotein envelope. Dropping that shield activates calcium channels, causing a massive influx of calcium right into the cell. This results in what we call hyperactivation. It's this asymmetric, frantic beating of the tail that physically prepares the sperm to penetrate the egg. It hits the turbo button, basically. Now, you might be wondering, in the vast, complete darkness of the fallopian tube, how do they even know where to go? Well, they don't swim blindly. Sperm literally smell their way to the egg. They possess specialized odorant receptors that guide them through a chemical trail. Even more incredibly, they use thermotaxis. They actively swim toward warmer temperatures. And get this, they are so sensitive, they can detect a temperature difference as tiny as 0.0006 degrees Celsius. Imagine being able to feel a temperature shift that impossibly tiny. It's mind-blowing. And here is a truly wild detail from our biological blueprint. Amazingly, that specific chemical trail the sperm follows, that invisible scent drawing them to the egg, smells quite similar to the lily of the valley flower. It's just a beautiful, strangely poetic little detail, hidden right there in this microscopic world. So, once that hyperactivated surviving sperm finally reaches its target, it faces one last massive hurdle, the zona pellucida. This is a formidable, thick glycoprotein fortress wall totally surrounding the egg. The sperm can't just swim through it. It has to actively break in. At this point, the sperm isn't just a runner anymore. It has to literally become a microscopic demolition expert. To breach this fortress, the sperm executes a fascinating mechanical and chemical process called the acrosome reaction. First, it physically binds to specific protein receptors on the wall, specifically the ZP3 receptors. This binding acts like a tripwire, triggering another massive calcium surge inside the sperm. That surge causes a little payload at the very front of the sperm's head to release specialized digestive enzymes. Think of them as chemical drill bits. These enzymes, hyaluronidase and acrosin, literally melt a hole right through the wall, allowing the sperm's membrane to finally fuse with the egg. But here's the really crucial part. The absolute instant that one sperm's genetic material gets through, the egg initiates the cortical reaction. Think of this as slamming the blast doors shut. The egg releases enzymes outwards that instantly harden that entire outer wall, making it completely impenetrable. Bam, locked down. This brilliantly ensures that only one single sperm can ever fertilize the egg, which keeps our human genetic math perfectly balanced, exactly 1/2 from the mother and 1/2 from the father. And just like that, we now have a fertilized egg, a zygote. Day 0 is in the books. But the journey isn't over, it just changes direction. Over the next few days, the zygote travels back down the fallopian tube toward the uterus. As it travels, it divides rapidly. 2, 4, 8, 16 cells. By day 5, it forms this hollow ball of cells called a blastocyst. And finally, right around day 7 to 9, it arrives at its home for the next 9 months, the uterine wall. This arrival is what leads to implantation. The embryo physically attaches to the uterine lining, essentially setting up its base camp. This exact moment of attachment is when a pregnancy is officially biologically established. But the endometrium isn't just sitting there ready at all times. It has to be specifically primed to receive the embryo. This requires high levels of the hormone progesterone, which is being produced all the way back in the ovary. With base camp established, the body's chemical command center takes total control. Estrogen steps in as the master architect here, orchestrating a massive remodeling of the mother's body. I mean, it doesn't just enlarge and strengthen the uterine muscle to hold the growing baby, it simultaneously prepares the breasts for future lactation, it acts as a roadblock inhibiting other hormones so no new eggs are accidentally released, it even physically loosens the mother's pelvic joints to prepare for eventual labor. It's a brilliant full-system override. But right at the very beginning, the absolute MVP of early pregnancy is a hormone called human chorionic gonadotropin, or HCG. So why is HCG so vital? Remember how progesterone was needed to keep the uterine lining intact? Well, normally, the ovary naturally wants to stop producing it and just degrade. But the newly implanted embryo instantly starts pumping out HCG. This acts as a massive survival signal. It travels backwards to the ovary, practically shouting at it to keep pumping out progesterone until the placenta is mature enough to take over. And we can detect this remarkable little hormone just 8 to 10 days after ovulation. And that specific window, that 8 to 10 days, is incredibly important for us, both medically and personally, because this specific HCG hormone is excreted in the mother's urine. It forms the exact biological basis for how at-home pregnancy tests actually work. It's our way of peeking into this completely invisible process. It's a really elegant illustration of how we harness biology for everyday diagnostics. When urine hits the strip, if that HCG survival hormone is in there, it binds to specially labeled mobile antibodies. This newly formed complex then physically travels up the strip. When it hits the test region, it gets grabbed by a second set of fixed antibodies, basically trapping the hormone in a little sandwich. And that trap, that's exactly what triggers the enzyme to create that famous visible line. A second line further up captures any leftover antibodies just to prove the test actually worked properly. Pretty clever, right? Which leaves us with a truly incredible thought to ponder as we wrap up today's explainer. We started with 2 microscopic cells navigating a massive, impossible obstacle course. And within a matter of days, that tiny resulting bundle of cells begins pumping out a hormonal signal so incredibly powerful that it stops the mother's normal reproductive cycle, completely reshapes her anatomy, and chemically demands the exact resources needed to build a brand new human life. Just how profound is that invisible chemical communication? It's a real testament to the breathtaking precision of human biology. I really hope you walk away today with a fresh appreciation for the epic, wild race that created every single one of us. Thanks for joining me, and I'll catch you in the next one.